Hello, everybody. Welcome to week 2 of our summer course together in human biology. I hope your first week has gone well. Moving into the second week. So we have 3 chapters this week. Don't need to say that because this is. Hi, Professor Powers here. This is the second chapter for this module. This is chapter 19, cell division. I am going to strongly request that you all use the reading assignment from McGraw-Hill to help you move through the reading. These PowerPoints are really to give you an overview of what's going on in this module, as well as some of the harder details. Okay, so here's the story. When we think about cell division, we have to go right back to that hereditary material of life. So here is a figure of a eukaryotic animal cell. And you see this nucleus. This is this purple structure, has a double membrane, has some nuclear pores here. And inside the nucleus, we have a nucleoid, which is this like darker purple area that they're showing. Purple, it's not really purple. And then you also see like some stringy kind of stuff all throughout this nucleus. This is called the chromatin at this point. That chromatin essentially is going to condense or wrap itself around proteins to become chromosomes. So you probably heard about chromosomes. For example, humans have 46 chromosomes, 23 that they got from their mother, 23 that they got from their father. But in a normal cell, during interphase, for example, we'll come back to that, but essentially this genetic material is all stringy. It's like a bunch of yarn just, pulled out of its skein and it's all over the floor. So that's what the DNA is going to look like inside this nucleus. And we call it chromatin. Okay, so the dilemma and challenge of a cell in cell division becomes, how do you get this stringy chromatin inside a double membrane bound nucleus inside of a cell with all these other things in it, the Golgi, mitochondrion, endoplasmic reticulum, and so on. And then all of that is surrounded by the plasma membrane. So how do we make 2 new cells knowing that the genetic material is packaged way inside this nucleus? So if you keep that in mind as I'm working through the topics of cell division, then I think that might help you go, oh, I get why that's happening. And think about it more intuitively rather than overthinking and trying to analyze. Okay. Now, I know that you all know what a chromosome is. Chromosomes, again, are this condensed DNA into a discrete structure that then the cell can use to help it with cell division. So the only time you're going to see chromosomes in a cell is when they are condensed and are preparing for cell division. There's 2 kinds of cell division in eukaryotic organisms. That's mitosis and meiosis. Coming back to that. A karyotype essentially is a illustration, if you will, of all the chromosomes that a particular person has. Now, for example, if I was having fertility issues and I've been trying to become pregnant, after serious trying for a couple of years, I might go see a reproductive endocrinologist or just go see an OBGYN. Essentially, somewhere in that process, they're going to want to analyze my chromosomes to make sure I don't have any translocations or deletions in any of my chromosomes, because that could aid to infertility. But what we see here is essentially the illustration of all your chromosomes. Now, I want you to look over here on the right-hand side for a moment. When our DNA condenses and it wraps itself around proteins, it's essentially going to be a single strand. Now, I know that DNA is double-stranded, so when I'm talking about single strand, it's a single, double-stranded DNA. What we see here on the right-hand side is a single double-stranded copy of DNA attached to an exact copy of itself on the other side. Okay, so when you see this in the replicated form, we call that a sister chromatid. Kind of makes sense. We need to differentiate between chromatin, chromosome. Replicated chromosomes are sister chromatids. Okay, so that's what we see here. The sister chromatids, one came from this person's mother and one came from their father, or the sperm and the egg, all the way through. So in a human karyotype, if you follow the numbers all the way down to the bottom, you're going to see that not only do the chromosomes get smaller and smaller and smaller, but they are numbered. Down here in the lower right-hand side, this is the 22nd pair. Right above it, you're going to see the pair of sex chromosomes. So this individual has one copy of an X and one copy of a Y. So that makes this person genetically male. Okay, so maybe you understand that. Let's take a look at one other thing. Chromosome 1 from mom and chromosome 1 from dad. They code for the same genes, whatever those genes may be. It could be height, it could be hair color, it could be genetic propensity for all kinds of things. We refer to chromosome 1 from mom and chromosome 1 from dad as being homologous or homologous because they code for the same things. Homo meaning same. Logous probably comes from the term loci, which comes from essentially where on the gene we have variations that code for that. Where on the chromosome we have the gene that has the variation that codes for something. So what I'm trying to say is chromosome 1 from mom and dad code for the same features. You just have different variations from each of your parents. Okay, so that now helps explain chromatin. Replicated chromosomes are called sister chromatids. When we start to pair up the chromosomes that code for the same features, now we have homologous chromosomes. Okay, so we have our chromosomes that code for our normal body features. These are called autosomes. So males and females have 22 pairs of autosomes. So that's coding for something other than your sexual characteristics. Then we have the 23rd set, which we said earlier is your set or your pair of sex chromosomes. You will notice there's one thing I didn't really mention. It's a little glob of protein, kind of like a big, huge polka dot here called the centromere. The centromere is always going to hold the two copies of that sister chromatid together. Okay, if you want to pause and get a little bit more information from here, then you can, but I'm going to go ahead and move on. This is essentially things I've already explained. So let's take a look at cell division. There are actually two kinds of cell division. The first of what we're going to talk about is the cell division of your body cells. Okay, so these are your normal everyday cells, epithelial cells, bone cells, your stem cells that make bone and blood and a lot of other things, cells that make up your fingernails, all along your digestive tract and so on. Okay, so we have two parts of the cell cycle. And when you think about the cell cycle, this is the lifetime of the cell. Like, what's it doing for a living? So the first part's called interphase. This is when your cell is doing something for a living. And then there's the actual cell division proper. So the way I like to think about this is a cell, when it's just been made, has a lifetime to live. And at some point in time, it is triggered to know, okay, I'm only supposed to live 48 hours or 2,364 hours. And then it is triggered to go into a number of events, if you will, so that it can prepare for cell division. Now, we know when we divide the cell, we need to, of course, condense that chromatin in the nucleus. This figure tells us a lot. I really suggest that you rely on these figures to help you remember things. The words are great, and it's things that I like kind of rehearse through my head when I'm trying to remember things, but it's usually figures that really help me. Okay. Interphase, all this purple area, even this part here, when a cell's kind of stalling in its life. This is called interphase. So what's going on? Here's my baby cell. It just formed from a previous mitotic event, and that cell's going to grow. It's got to gain its Golgi. It's got to make sure that it has enough lysosomes. It's going to increase in size, so it's going to gain cytoplasm. But then that cell is triggered to go ahead and go through the DNA replication phase, okay? Because the cell needs to replace itself before it gets to the very end of its life cycle. So all of the DNA, that chromatin, is going to replicate. So that's a whole nother process called DNA replication, which we're not going to address in this class, but it's a pretty cool, fascinating thing. You can find some really great videos on that out there in the world. Okay, so we call the S phase. S for synthesis. It is synthesizing DNA, making DNA. So growing, G1, S, synthesis, G2, continuing to grow, and then you see the final preparations for cell division. Then the cell is going to go through the actual cell division process, which generally speaking, we will refer to as the M phase. And the M phase is going to include the subphases, prophase, prometaphase, metaphase, anaphase, telophase, and then finally, this last part, cyto means cell and kinesis means divide. Now we have essentially two cells. So we need to talk about that process. Okay, so mitosis and cytokinesis. Mitosis is essentially when we are literally dividing the copies apart, and we want to make two identical copies when we're done. We start with one original cell, and that one cell is literally going to donate itself, if you will, to the cause. So that's where in the nucleus, the chromatin is going to replicate itself. Then we're going to see that those chromosomes in the replicated form called sister chromatids are going to appear with the centromere holding the two identical strands of DNA together. Then it's going to separate those strands, and eventually we have to pinch apart and separate the stuff that's in the cytoplasm. So we have mitosis and cytokinesis. So what you should know is that at the same time that we're dividing cells, there's also what we call programmed cell death. So this is particularly fascinating early on in life when you're going through cell division of the embryo. And to make what would be the characteristic morphology or shape of that particular body form, so if it's human or if it's whatever it is, you might have some cell death so that where the cells are replicating, they are maybe pushing out and creating tissues, and somewhere else the cells are dying so that we maybe like curve the tissues, okay? Also, we know that apoptosis or programmed cell death happens when cells are excessively damaged. All right, so now let's go back just for a moment and let's take a look at essentially some checkpoints. What you see here is these stop signs. What you should know is that there are checkpoints during cell division to ensure that everything is going as it should. So when we think about cells unnaturally dividing or overly dividing, we're often thinking about situations such as cancer. Okay, cancer essentially is unregulated cell growth. It's where these checkpoints are not being reinforced. So essentially what we have is we have some cells dividing out of control. So you can pause and take a look at this. This is more for information and not so much about you need to know all the checkpoints. Okay. All right, so let's take a look at mitosis proper. So we know that mitosis is occurring when that sperm fertilizes the egg and we have the zygote. That one single cell zygote is going to turn into a multi-million celled fetus at some point and 100 trillion cell adult at some point, and that's all because the cells are dividing to make copies and to make copies and to make copies and to make copies. For us as adults, we know that we're replacing old cells. If you've had surgery, it's responsible for healing. Sun damage that you might get, suntans or whatever, we know that our skin is sloughed off, replaced by cells during mitosis, so our new cells come in. So a couple other things, we start with the parent cell, it makes daughter cells, and in general, we're going to be talking about organisms that are diploid. have two sets of chromosomes.